Yeah. So, you remember what we discussed is that RAS kinase pathway can be activated by anything upstream, like your receptor tyrosine kinase is activated, or your cytokine receptors can activate it, and many other pathways can activate the RAS kinase pathway, right, RAS MAP kinase. So, here what we will take a specific example where a G protein coupled receptors are going to activate RAS MAP kinase pathway, and this specific example is in context of yeast mating. You guys have heard that sometimes under stress this yeast will undergo, usually the budding is they will undergo division by budding, but under specific conditions they can undergo mating as well. So, when we talk about this, essentially this is what is shown over here, right. So, there are two types of yeast, haploid yeast cells, one is A type or one is alpha, and these are the mating types what they call, right. So, the important part is that the A type will secrete A factor and alpha type secretes alpha factor, which is nothing but the pheromones which is required for the process of whatever reproductive whatever mating is going to happen. For that process they secrete these pheromones. So, this A type secretes A factor, alpha secretes alpha factor, and the important thing is that these will go and attach, they have specific receptors. The alpha factor, the A type has a receptor for alpha factor, whereas alpha type has receptor for A factor. That is like, it is like complementing each other. And similar to GPCR pathway, this pathway also operates where these acts as a ligand, goes and binds to the receptor a GPCR, and this GPCR will be carried out, the signal transduction is carried out by the GPCR pathway. Something to make a note of it which I missed to label over here. Do you guys remember what happens in a typical GPCR signaling? Whenever G ligand goes, binds to GPCR conformational change happens, goes, activates the GTP, G proteins, right, and G alpha and G beta gamma, there are two complexes where G alpha is associated with membrane, G beta gamma is associated with membrane through gamma complex, right, and both. So, the moment these GTP GDP is exchanged with GTP, G alpha gets activated and the GPCR initiates the interaction, right, they dissociate. And who is the factor which goes and induce the signaling cascade here? G alpha, right. In this particular example, remember it is not the G alpha, it is the G beta gamma which is important to initiate the signaling cascade. This is very specific example that there might be another examples of similar pathway in yeast mating. During the yeast mating, when the GPCR will be bound by G alpha factor or A factor, whatever respective factors are, there is a signaling cascade which kicks in where the G alpha beta gamma, right, G alpha and beta gamma, when these two are together they get separated, but G alpha does not have any specific role. G beta gamma is the signal transducing unit in this particular context. Remember that, which is different than your canonical GPCR signaling. No, GTP will be with to G alpha only, but G alpha will dissociate, and so in canonically G alpha will dissociate, go and will bind to, let us say, adenylyl cyclase. In this case, this guy will not go anything, it will be remain membrane bound. G beta gamma will go and bind to downstream molecule. So, we will see the pathway now. So, like this is the pathway. So, now in yeast you will find every equivalent enzyme which is there in this RAS MAP kinase pathway, which is similar to the mammalian system. Okay. So, I have given the equivalence of what is what here. Can you guys see this? So, just focus on this mating pathway right now, we will go to the other pathway after some time. So, this left side pathway, please focus on this here. So, you have receptor, this is your mating factor comes and binds to this, and this is your activation of G proteins will happen, right. The moment it happens, your G beta gamma and G alpha will segregate. G alpha is no more in picture any required now because it is not signaling entity, whereas G beta gamma will go and first associate or recruit this STE5, which is a scaffold protein. I would want, please pay attention to this STE5 scaffold protein. for that matter the scaffold protein, because that is going to be part of some discussion we will have some other time. Why it is called as the scaffold protein? Because it is going to provide a platform to build or to recruit so many other downstream signaling competent kinases on this platform. Okay. So, G beta gamma will go and associate with your STE5 scaffold protein. Remember that these STE5 scaffold protein then will lead to the activation of your CDC24. What is CDC24? It is equivalent of SOS. What is SOS? Son of 7 less, that is your GEF, which goes and activates CDC42. What is CDC42? CDC42 is equivalent of RAS, which happens your canonical RAS pathway in mammals. Once the CDC42 gets activated, you know, when your RAS gets activated, what gets phosphorylated? There is a kinase which is RAF kinase, right. So, in between there is this STE20 kinase, which is a MEK kinase. It goes and phosphorylates and activates this STE11. Now, you can see that STE5, which is the scaffold, all the kinases are being built on the same scaffold, means it is acting as a surface where multiple kinases can go and attach and they will phosphorylate each other. So, it is kind of a signaling hub that is being generated over there. It is a very typical example and very interesting one, where the upstream signaling is leading to the active recruitment of the scaffold protein first and the recruitment of RAS equivalent, which is CDC42. And this, remember, this is happening in yeast mating pathway, where the mating factor is going and activating a GPCR equivalent pathway, right. The only difference is G beta gamma is the signaling competent authority here, not the G alpha. Then this STE11 goes and phosphorylates STE7, which is equivalent to your MEK kinase, and this STE7 goes and phosphorylates first 3, which is nothing but your MAP kinase. Again, this first 3 similar to MAP kinase enters the nucleus, it goes and phosphorylates these two guys DIG1, DIG2, so that it will not inhibit the transcription factors and which lead to the transcription of mating type genes. You understand what is that? So, the pathway which we saw some time before over here there was a specific example we saw. What was the example? When you withdraw serum and when you add serum back to the system, what is happening? There is a induction of early response genes. CFOS is one of those early response gene. Where does this transcription factor go and bind to? It goes and binds to the serum response element, right. So, your MAP kinase gets activated, goes and phosphorylate this particular TCF, which is a transcriptional co-activator, which goes and associate with SRF, which is serum response factor. These two guys go together and sit at SRE, then they drive the expression of FOS, right. This is kind of feed activation, everything is happening. In this case, what is happening is you want to the way this MAP kinase pathway is going and activating is phosphorylating these inhibitors. So, there are some transcriptional, what we can say, the repressor kind of, right. So, these two guys are repressors which are repressing this pathway when pathway is inactive. The moment pathway is activated, your first 3, which is MAP kinase, phosphorylates these two guys DIG1, DIG2. And the phosphorylated DIG1, DIG2 will not inhibit the pathway. So, STE12 and MCM, which are the downstream transcription equivalent factors, they will lead to the transcription of mating type genes. Did you understand, Pratibha? No? Shall I repeat? Okay. So, when pathway is not active, see, mating is not, in yeast the mating is not happening regularly, right, which means that you need not to have those genes expressed normally, right. So, when you do not want something to be expressed normally, what do we do? Normal cells, they will undergo proliferation, right. So, when a cell is undergoing proliferation, which pathway will be on and which pathway will be off? जोर से that is on. Which pathway will be on? Off. Apoptotic pathways, they should be off because you do not want to die, right. Everybody agrees to that. Similarly, when you do not want to have mating, what will yeast do? Will it express the genes which are required for mating? No, right? So, when pathway is off, when there is no mating factor which is going and activating on your GPCR, pathway will be off. In that case, how do we keep the pathway shut? Sanjit, what do you think? How can? You have transcriptional repressors. These repressors will go and occupy the elements. That is what is happening. This DIG1 and DIG2 are repressors. They will be sitting over there. So, they inhibit the transcription of mating type genes. When pathway is activated, what happens is your MAP kinase will come and that will phosphorylate these two molecules. When these two repressors get phosphorylated, they will be removed from the nucleus, they will be removed from the response element or the promoter, where the active transcription factor can go and drive the expression of mating related genes. Is it not clear? Clear? Is ECPC, right? So, this is one example how a particular signaling cascade is being carried out.
So means here the phosphorylation is not for activation but
It's
for
in,
inactivation.
it is kind of indirect activation of transcription program by inactivating the negative regulators. Double negative is positive, no, like that.
So the repressor which has to be stopped means the phosphorylation is needed for that repressors.
Yes, phosphorylation is needed to drive the expression. दुश्मन का दुश्मन दोस्त होता है type. You understand, right? Now, this is one example, right? So, if you guys recollect, if you go and read in general literature, you will find that there are so many stimuli, there are so many pathways, so many extracellular signals, they go and activate these MAP kinases. And this MAP kinase, RAS MAP kinase pathway will be activated. Now, the question is, you remember here we saw in this particular case, you have RAS which goes and activates RAF gets activates MEK activates MAP kinase. Now, there has to be a specific context to the activation of MAP kinase, right? Not any other signaling factor cannot go and activate the MAP kinase randomly. So, for example, so this MAP kinase which gets activated will go and activate the transcriptional program, right? We saw two examples. CFOS example you take. CFOS is the one when your cells are already starving and then suddenly you give them food. Now you want to grow, you want to proliferate quickly, right? In that case, you want to switch on all the genes which is required for the cell proliferation. This is one type of response, right? When you are starving, you get food and you want to drive that. In case of mating type, here there is some limitation, that is why you are undergoing mating type of this kind of, you are following this pathway, where there is a external stimuli which is coming, whether it is CFOS activation or through CFOS activation or through GPCR dependent mating type activation. In both the cases, who is getting activated? Sorry, when you activate, when you want to drive the expression of early response genes, or you want to activate drive the expression of mating response genes, who is the molecule which gets activated from the upstream signaling? MAP kinase, right? So, this MAP kinase should know why it is getting activated and who is activating it. You understand what I am saying, right? There has to be a context. वो MAP kinase cannot get activated randomly. वो mating factor से activate होके early response gene को transcribe नहीं कर सकता. That is not right. You understand, right? So, you are following what I am getting at, which means that if this MAP kinase has to do a specific job, meaning this particular signal has come and I have to drive the expression of this in response to this, not to something else. So, let us say there is X signal which leads to the cell proliferation, there is a Y signal which leads to the, let us say, induction of apoptosis. At the end of the day, MAP kinase is doing it. Now, MAP kinase should know when to activate, even if X does it, it will not gonna go and activate the apoptotic genes. So, how does it differentiate the upstream signal? What is that thing which is going to help it to respond to the upstream signal correctly, so that it can go and give correct transcriptional response, right. So, the one yes. So, the example of that is your osmoregulatory pathway, which is again studied in yeast, and that is when I say that pay attention to this scaffold protein. Now, you can see that the receptors are different, but and the scaffold protein is different, but rest every other pathway is similar, no, early response pathway is similar. Like you see there are these two guys, SHO1 and MSS2, these are this something whenever you give osmotic shock to yeast cells, right, whenever the extracellular solute concentration is changing, this yeast cells will respond to that, right, they will have to sense that. So, there are certain receptors they will sense, these receptors will sense that, right, but what happens, they will again recruit a particular scaffold protein, they are not going to recruit the STE5 scaffold protein, because STE5 scaffold protein was very specific to the mating factor dependent GPCR activation. They recruited another protein called PBS2, which is another scaffold protein. Again, however, the initial activation is similar, CDC42 gets activated, STE20 gets phosphorylated and activated, this STE20 goes and activate and phosphorylate and activate STE11 as well, STE11 goes and phosphorylate and activate MEC as well. So, the initial pathway is same, the upstream signal is different. Now, this MEC will go and activate another kinase, in this case there was first 3 which is equivalent to MAP kinase, here there is another MAP kinase equivalent which is HOG1. You understand what I am saying? So, MAP kinase is not there is not a single kinase, there are multiple, it is a family of kinases, right, with like P38 MAP kinase, you must have heard in mammalian systems, no, people talk about P38. Whenever we talk about MAP kinase pathway, this is the commonly used P38, or as you were talking about, we check ERK or phospho-ERK, right, that is what we call, this is MAP kinase, P38 is MAP kinase, right. So, in yeast, and there are others, so there is one more error, I missed it, I will remember and I will let you know. So, in this case, even though you are, so if you remember, you have RAS, then you have RAF, then you have MEC, and then you have MAP kinase. This is a canonical pathway. Even in this case, whether it is mating or your osmoregulatory pathway, same cascade is happening, RAS, RAF, MEC, right. In both the cases same pathway is getting activated, no, but what is different in both the cases is your scaffold protein, you guys are following, and this scaffold protein, the end product, the end phosphorylation, here it was FOS3, no, FOS3, which FOS3, sorry, and here you have HOG1. So, this will go and drive the expression of mating type genes, this is going and driving the expression of genes which is required to provide protection to against osmotic shock or osmoprotective genes. Is it clear? Everybody understand what? It is not a part of scaffold, it is another MEK enzyme, that is kinase which is recruited. It is not a part of the scaffold. Scaffold's job, it does not have any catalytic activity. Scaffold job is only to provide a platform where any other molecules can be organized. That is the general function of scaffold proteins. No, that is wrong. So, some of these things in this book, no, there will be, so this is I am following Lordish book, so the image is taken from there. but I do not think it is the part of that. Scaffolds do not have catalytic activity. Scaffolds will not have, that is why it is called a scaffold protein.
So in both the cases, MEC are same, but they are activating different MAP.
Downstream substrate is different, and that is when your Down's response is also different. That is when you could go and activate this osmo responsive or other targets will be there. So, these other targets will be some of the transporters, some ion channels, ion transporters, those genes will get activated. These are not necessary that it always has to go and phosphorylate the transcription activator, right. It can go and activate some other proteins that can get recycled back to the membrane and start doing their job. Is it clear? So, in general, you guys remember when we talked about your cyclic AMP response related genes and how do we differentiate the signaling cascades, right. So, there was the role of A-cap proteins we read, no, you remember? A-caps are also nothing but the scaffolds only, right. They will hold your, what is that, pKa, and they also they provide a specific localized action of pKa and what not. That is how you can build up your signaling repertoire of multiple, respond to different signaling. Similarly, these scaffold proteins are also equally important. Is it clear? And something to make a note of, these kind of pathway which is elucidated over here, right, ST11, ST7, FUS3 and what not. So, essentially what people did in East is that you knew that how do you deduce these new molecules? Yeah.
Sir, the names of this like CDC24, CDC42 are original names, but they are functioning like SOS or RAS in in the mammalian, and if you are talking about yeast, so the main names are CDC24 and CDC42, which is maintaining the activity of SOS.
I think these pathway was initially discovered in Drosophila. So, the names come from there. So, what you do is you find the equivalent of them in other systems.
So, in this here the equivalent of SOS is CDC24.
Correct. So, I just wrote it ulta because CDC4, we are studying the yeast pathway, but I wanted to tell you guys what is CDC40 equivalent in mammalian system. Is it clear? So, what people did it, so this particular pathway is very interesting, right. So, what you do is you have this ST11 or ST7, how do we know that these proteins do function in this pathway? Here what you do is you take a yeast haplotype or haploid yeast where everything upstream is working well, but still they are sterile, right, meaning everything checks out. But if you remove your ST11, none of the pathway is going to get activated, no, because that is the first responsive kinase which is coming. Or if you remove the scaffold protein, again still they will still remain sterile, right. So, that is how, so in yeast people carry a lot of genetic screens. You know why? Have you heard about genetic, I mean nowadays people are not doing it as much, how there was an era of genetic screens. They provide lot of advantage over there, but they are haploid, which means there is single copy. So, if you remove that, definitely whatever phenotype you are going to get is because of that gene. So, people have done all these initial discoveries, DNA damage, cell cycle, falana, dhimkana, trafficking, autophagy and all that is studied in yeast. So, it's a very powerful model system. Nowadays it is dying. I mean, Anushka is not here, but their lab works on yeast, but nowadays you will find very few people trying to use it, but it has done its job very well. So, that is how they have been discovered. So, if you remove ST11 by any random mutagenesis, and if you try to see whether that particular yeast is going to be mating competent, and if you find that hey it is not, then you go back and find what went wrong, and then you identify hey, these genes might be important. Now, the question is this ST11 where does it stand, right? And if you remember last time what we said, in this pathway even everything upstream is gone bad, and if you overexpress MAP kinase, it could still rescue the phenotype. So, in this case, in this yeast, even if your ST11 is bad and if you overexpress MAP kinase, that can still rescue the phenotype, which means that this could be upstream to MAP kinase. If you remove MAP kinase, everything else is there, which means and still they do not rescue the phenotype, then we know that this is at the end. This is upstream, downstream, accordingly you can do it. This is how you built up the signaling pathway. I do not know in your PhD thesis whether you need to do that ever, but you have to do, right. So, you have protein A, protein B, they are functioning somewhere, you want to build where does protein A come from, or when does protein B function, whether upstream to A or downstream to A. That is how you do, no. So, you remove A, try to put B and you see whether it rescues the phenotype. If it does not, then you put both of them together and you see whether they give additive effect or not. So, that is how you build the signaling cascades. Is it clear?
So what is that MEQQ?
It's MEK kinase. So, it's a kinase which is going to go and phosphorylate another MEK. Like this MAP, no, MAP kinase is there, right. So, like that, these are named like this is super family, but they will go and phosphorylate some other MAP. So, that MAP can go and phosphorylate some other MAP kinase family member. So, that is how you build that MAP kinase pathways. Is it clear? Okay. So, this is where the MAP RAS minus kinase pathway is over. So, I will just try to tell something about this hippo signaling. It is very new to me also, and it's very interesting, and it has wide studies, people are studying it. It is not very old pathway, it is discovered in 1995 in Drosophila again. I do not know, I read it somewhere where they say that this particular mutant led it to the hippopotamus kind of phenotype, that is why they called it hippo. I do not know, do not quote me anywhere. So, this is how it looks. You have wild type Drosophila, you look at the head of Drosophila, it looks pretty good, right, but look at this. This is the mutant hippo pathway gene, and that is how the massive outgrowth, tumorogenic growth or anything happens, and that is how these genes are named hippo pathway genes. So, this is what it is. In 1995, they actually discovered this hippo pathway, and over a period of time, there have been lot of discoveries till recently, and we will see how the equivalent of Drosophila and mammals. So, it is very complicated pathway, but again, and it does too many things. Maybe it's difficult to understand each and everything what it is going to do. So, this hippokinase HPO is the kinase which is the molecule of importance over here, which is in Drosophila, and the equivalent of hippo is your MST1 and 2 kinases that are present in mammalian system. So, this is what the comparison we are doing. So, hippo goes and phosphorylates this SAV, which is equivalent of SAV1 in mammals. The downstream of hippo is this WTS. Their names are also very weird. I do not even remember this. VRITS or something is WTS stands for. That is the molecule which is very important, and this can get phosphorylated. Then there is another cofactor, or the molecule which functions with WTS is MATS, and the equivalent of this MOB, and LATS1, LATS2 is equivalent of this WTS. So, LATS1, LATS2, these are mammalian. So, there have been, these all are kinases. So, this MOB1, MOB2, they are also kinases. So, this MST1, MST2 goes and phosphorylates these kinase gets activated, goes and phosphorylates another transcription molecule. So, this SKI YKI is called as SKI, it is a transcription factor, that is the molecule of your importance, right Muskan. So, this molecule gets phosphorylated, and whenever this molecule is phosphorylated, it remains in cytosol. That is means when the pathway is on. So, this is again opposite happens. When pathway is on, something gets degraded. When pathway is off, it works functioning normally, right. So, this table it's maybe it's not readable, you guys will have access to this. This gives the equivalent of Drosophila and mammalian and their functions, like hippo MST12, it's a kinase, and what is the conserved domain is present in that, and then there is MATS is MOB1 to Yorkie, not SKI, Yorkie is YAP-TAZ pathway, which is S associated factor. YAP is S associated factor, and it has its friend which is called as TAZ, and this YAP-TAZ, these two guys are together always, they go and do the job downstream. In fact, something which is very important, this YARKI, YKI or YAPTAZ, they themselves could not go and activate the DNA transcription. They do not have transcription activation domains. So, they always go and associate with this factor called SD in yeast, whereas TEAD in mammals, and then they drive the expression of downstream genes. It is very important to make a note of it. Okay. I might not ask questions to you guys about hippo pathway, this is additional learning.
So these all are kinases?
Yeah, these all are kinases. So, YARKI is not a kinase, but upstream everything is kinase, like this hippo is a kinase, then there is VRATS is another molecule that is a kinase which goes like LATS12 is a kinase, MOB12, I think these are again kinases, MST1, MST2 is kinase, and then you see this MAP4K kinase, this is another kinase that can act in parallel with MST1 to activate LATS kinase. Similarly, this HAPPY or MSN, that is equivalent of this MAP4 kinase, that can go and in parallel to hippo it can activate this pathway. And then there are other molecules like Kibra and what not, these guys are also can go and activate the pathway. So, it is little bit very complicated. I will show you one chart where you will realize that it can get activated by anything, it looks like to me, and it has so many functions. And so, these are the equivalents of this. So, this is how it is when pathway is on, which means that YAP is inactive. So, you can see this little bit. So, hippo activity goes down, YAP activity goes up. That is how the pathway is. It is antagonistic to each other, it is opposite. So, whenever hippo is on, what happens is this MAS kinase will go and activate LATS, LATS will go and activate MOB kinase, they will together will phosphorate this YAP. So, there are two ways of sequestration of YAP. YAP will either get sequestered in cytosol by some 1433 family proteins. You remember 1433 family and some actin cytoskeleton molecules. So, like this 1433, sorry, this phosphorylated YAP gets sequestered by 1433 family or it gets degraded. So, there is no transcription happening. When pathway is off, so what happens is that whenever pathway is off, this none of the upstream kinases will be activated. So, essentially YAP is very happy to go into the nucleus and drive the expression of so many genes which will have these many functions. And something which is very relevant to this pathway is that this pathway has been extremely studied in case of context of cancer, especially liver cancers and what not other cancers. So, this is what I was telling you guys. If you look at it, it can get activated by stress signals, which is nothing but hypoxia. You guys understand hypoxia, right? It is low oxygen, where the VHL and those guys have been very well studied. William Kevin Jr. was one of those person who discovered this VHL factor. He was awarded Lasker, I think he will get Nobel eventually. So, then there is osmotic stress and what not, or it can go through cell polarity, you know, right, these apical basal polarity pathways, they are important. So, again in each of these there are other 4 or 5 different molecules which will go and activate each other or inactivate something. Whenever there is some liquid flow stress or in cell density dependent pathways, and there is soluble factors which are GPCR dependent. I will give one example. In this example, you guys do 2D cell cultures, right? This has been studied in 2D cell culture. So, the proliferation rate of cells is negatively correlated with density in the monolayer culture. That is what we culture, no? Some of you might be doing some 3D cultures as well, but in 2D cell cultures, you know there is something called as contact inhibition, right? Now, at high confluence of mammalian cells, which leads, so people have studied that whenever your cells are at high confluency, it leads to the activation of LATS kinase. This LATS will go and lead to the phosphorylation and inactivation of YAP. Furthermore, the overexpression of YAP, it could reverse the growth inhibition. That is why it shows that YAP is downstream than LATS. So, this is the pathway whenever you have cell density. One of the complex which is known to go and activate this YAPTAZ latent function is this Crumb's complex, which is where there is a function of these molecules called AMOT, and so these are too many names to remember. I am really sorry, I do not remember all of their names. So, other pathways I have heard their names some of the other way, because during you one or the other time you come across them. This is one pathway which I never worked with also. So, I do not connect with these names as well. I remember in my PhD lab there was somebody who was working on this LATS kinases, and that was hippo pathway, and that that project was not successful. That is another reason that I definitely don't remember it. So essentially this crumbs complex which goes and activates this, leads to the phosphorylation of this YAPTAZ, and that's how the pathway will be regulated. So essentially this is one example people have, which is very routinely studied in your 2D cell culture. So that is one reason when, and this is very important for the maintenance of the tissue, growth of the tissue. It's not for the proliferation always. So your, after some times when your soul cells have grown enough, the number of cells is enough, the cell has to increase in size, right. So every tissue will grow in certain size, no? There is some shape and volume, and this thing is that. What pathway is that decides that this is enough for that tissue? This is this pathway, hippo pathway. If it is not there, then it will go uncontrolled. That's how the important this pathway is. I do not know why it was not there in that book. I should include it next time.
Because the head looks like a hippo, it is called hippo pathway.
Yeah, so I mean that's somewhere I read, but I exactly don't know whether it's right.
It can occur in a young individual also.
In humans,
Adult humans.
I do not. In cancer, so this pathway is, in cancers it is deregulated, right. So that is no, hippo doesn't happen, but uncontrolled the growth of a tissue will happen. So in that Drosophila head also it was uncontrolled growth of the tissue. It's
It was seen in head.
it was seen in head. So it looked like that. So but in humans, I don't think humans will become hippos. So this is another example, the role of hippo pathway in your liver regulation. So if you take, this is your control liver, and whenever you overexpress or activate YAP, you can see the liver is increased in size, right. That's what happens. It's almost 4 to 5 times bigger than what the normal liver looks like. But if you down regulate YAP in these contexts, you see there is a reversion of liver size has happened. Whereas if there is consistently in, this is mouse, I think this paper is about mouse. Human का कुछ नहीं है. Human में it's very difficult to do that, no, because you will need 1,000, 10,000 permissions, and who will donate? So mouse bichara hai accessible. Nowadays there are so many people who are anti-mouse also, no? They are saying you should not use animal models for studies. So right. Right, and so if you have persistently activated YAP, you can see, now you can see there are some small, these arrowheads show you the nodules which are nothing but the cancerous growth which is happening. Now this is in hepatocellular carcinoma, that's what is happening. So your liver is increasing in size, but it leads to the hepatocellular carcinoma as well if you have consistently hyperactive YAP, which means the pathway is off, but YAP is hyperactivated. Is it clear? So I did not include too much material. I could, this is what I can give you, Aniket, not more than this. But the slides will be shared, so you can go back, and there is this review which I have cited from this one, not this one, signal transduction and targeted therapies. You can go through that if you are interested, and you will get more information. This is about your YAP, TAZ and hippo pathway. So with this, we finish what we are supposed to learn in this course. I think we almost covered very important signaling pathways.